It is time to kick off our afternoon sessions. Our first science session here in Regency ABC is Somatic Instability. This session features two speakers and will be moderated by Dr. Vanessa Wheeler from Massachusetts General Hospital. The second science session on gene editing will follow directly after. In Regency EF, which is just to my left over here, will be the HD care session on trauma-informed care. After this session, there will be a 15-minute break. Now please join me in welcoming Dr. Wheeler. Come on up.
Thank you so much. Thank you to the organizers for this opportunity to talk to you today. So this is going to be a kind of dual science session. I'm going to kick off and talk about somatic instability, and Ricardo is going to carry on and talk about CRISPR editing, and there are overlaps with the two. And feel free to interrupt. I think it's meant to be a sort of fairly informal-ish session, so if you have any questions, so it might be hard to see you, but okay, so okay, these are my disclosures. And so I want to start with this kind of fundamental slide about Huntington's disease, which is that the CAG length that a person inherits drives the timing of disease onset. And so the longer CAG you have, the earlier age of onset. This is the strongest single determinant of the age of onset. Sorry, I'm coughing, and so is Ricardo coughing, so we're going to be a pair. And obviously you can see from this that for any given CAG repeat length inherited, there is a wide range of ages of disease onset, and then the subject of genetic modifiers, which I'll come to. So the CAG repeat length is highly unstable, as we know, and I just want to point out this a plug here slightly for a chapter that we contributed to in this book of Huntington's disease, which also has a lot of other chapters about all aspects of Huntington's disease, so it's very interesting to read. And the chapter that we wrote was about somatic instability, or instability in general, and this shows as you're aware, I'm sure, that the repeat is highly unstable intergenerationally, so in the germline, and in transmissions from fathers, the repeat tends to dramatically expand. In mothers, it's unstable as well, giving rise to expansions and contractions. So this underlies the repeat length variation in patients that allows us to then correlate that with phenotype. So as has been described for a very long time now and put really into the forefront here in a paper from Peggy Shelbourne in Scotland many years ago now, is that there is also extensive somatic expansion of the repeat in HD postmortem brain. And this shows an example of an individual, two individuals actually, and you can see the huge repeat expansions, and in an individual on the right here, prior to pathological cell loss, expansions that went up to even 1,000 repeats. So the repeat is highly unstable in the brain. We also showed a number of years ago, thinking about the variation in age of onset that's associated for any given repeat length, what might underlie that variation. And the hypothesis was that perhaps individuals who had earlier onset may have more expansion in their brain than individuals who had later onset. And indeed, we showed this, and we found that there was an inverse correlation between the degree of somatic expansion in the brain, higher somatic expansion in the brain, and an earlier age of disease onset for a given repeat length, so controlling for the repeat length that you inherit, suggesting that individual-specific factors might contribute to the age of onset. So we've also gone on to show that somatic expansion is tissue-specific. This shows a study over a range of brain and peripheral tissues, high in the brain and much lower in general in peripheral tissues. And also interestingly that within any one individual, although the absolute levels of instability differ between tissues, they are correlated between the tissues. So, for example, between BA9 here, cortex and cerebellum, there's strong correlation between in the instability, and also between blood instability and brain instability, suggesting that the instability in blood for an individual may be a surrogate for instability going on in the brain. So more recently, we've learned a lot more about instability in patients from really great work done from the labs of Steve McCarroll here on the left and Nat Hines on the right. Steve McCarroll has done single nuclear RNA sequencing, where he's been able to develop a technology to measure CAG repeat length in individual cells and of many different cell types. Nat Hines' lab took an approach of enriching for different cell populations in the brain. And both of those studies have shown that the medium spiny neurons in the striatum exhibit extremely high levels of somatic expansion compared to other cell types. So just, I'll talk a bit more about the most recent GWAS data, but as you're all aware, the genome-wide association studies for modifiers of onset, the first paper was published in 2015, and there have been a number of subsequent iterations of the GWAS with progressively increasing sample size and also looking at other phenotypes. The GWAS have really validated now somatic expansion as a key driver of the rate of disease onset. And so there are a number of key findings that came out from the first couple of papers. So as the patient size began to be increased, there was increased power to detect effects of these non-canonical alleles. So the canonical Huntington allele has a CAA-CAG, the penultimate codons of the repeat. And so there are rare non-canonical alleles, either with loss of interruption or where this CAA is duplicated, so CAA-CAG is duplicated. And being able to look at those in this study, in the GWAS, showed that allowed you to distinguish the CAG length from the glutamine length that was encoded by both the CAGs and the CAAs, and showed that the pure CAG length was a better predictor of the age of onset than the total glutamine length. So that was one key finding from the GWAS. The other was that many of the onset modifiers were DNA repair genes already known from studies in mice to modify the repeat instability. And then the other thing which was known was that somatic expansion itself is CAG length dependent. So putting all these together, the hypothesis that has emerged now is that the property of the CAG repeat length driving onset that I showed in the first slide is its propensity to expand in somatic cells. So I'll just show the most recent GWAS. So this is currently on bioarchives. This is from the GEM-HD consortium. And so this is a GWAS of about 12,000 individuals, mostly of European origin. And I'm showing here two different phenotypes. One is age at TFC6, and the other is age of onset. And they're just mirror image Manhattan plots. And so here I'm highlighting several loci and genes that are in red, which are implicated in DNA repair, and which are DNA repair proteins and implicated in somatic instability, as well as a number of other genes in blue, which do not have obvious functions that implicate them in that process and may be involved in other aspects of the pathogenic process. The other The other part of the most recent GWAS was extending the spectrum of clinical phenotypes that were analyzed. I'm not showing all of them here, but I just want to compare just what the Manhattan plots I had before, showing age at TFC6 and age of onset, with a GWAS for expansion in blood. This was done using a MiSeq assay, really nice MiSeq assay developed by Darren Monkton's lab, and directly measuring repeat expansion in blood and looking at the modifiers of that molecular phenotype. And so they're sort of piled on top of each other. And as you can see, many of those DNA repair genes that are found to modify onset or clinical or TFC6 here also are found to be modifiers of expansion in blood. So those are all the red ones. Interestingly, so none of the blue ones came out as modifiers of expansion in blood. And interestingly, there were some additional genes that emerged. So for example, MSH2, MSH6, a SNP in the 5' UTR of Huntington, as well as MLH3, which was close to significant for clinical phenotypes, but genome-wide significant for expansion in blood. And so this distinguishes effects of these modifiers in blood, modifying stability, and in the brain cells driving disease clinical phenotypes, suggesting that the way in which they act subtly may be different in different cell types. So I talked a bit about the non-canonical repeat structures that allowed in the initial GWAS to distinguish the effects of the pure CAG repeat from the effects of the encoded glutamine tract. And so I'll just talk a little bit more about these and what they are doing. So these are the structures. So on the top is the canonical repeat. So there's a pure CAG repeat followed by a CAA CAG. So that encodes the glutamine polyglutamine stretch, and there is then a polymorphic CCG stretch in kind of purple, preceded by a CCG CCA, and all that encodes a polyproline tract. There's a CAA CCA loss allele. That's the most common CAA loss allele, where both of those, the green and the yellow, are lost, and so you have a pure CAG repeat and a pure CCG repeat. So that's known as CAA-CCA loss. And then there's the dupe allele, where the CAA-CAG is duplicated. And so when you correct for the number of pure CAG repeat lengths, and then you ask, what do these alleles do? And so this is the impact on age of onset relative to the canonical. So for the CAA-CCA loss allele, so it's a strong disease-hastening haplotype, okay? And so you might expect, if this works through somatic expansion, that it would be associated with much higher levels of somatic expansion. But in fact, it's not. In blood, at least, the impact on repeat instability is to slightly reduce instability. The CAA dupe allele is associated with delayed onset, and it doesn't have any impact on blood instability. So this suggests that these two alleles, they're very interesting, may be affecting the disease pathogenesis through mechanisms other than somatic expansion. So broadly, and maybe sort of simplistically, the GWAS kind of implicates two broad categories of modifiers. One of those involved in repeat expansion, and the other that may be involved in a sort of broadly toxicity process or processes, which we don't really know, I'd argue, what they are, and some examples are shown on the right of what that toxicity driver could be. And of course, this is a bit simplistic because we don't really, some of those actually may be involved at some aspects of repeat expansion, sort of indirectly. And the other new data that's arisen here, this is now data from Steve McCarroll, his single-cell study. And his single-cell study has now suggested multiple phases of pathogenesis, and potentially then different opportunities for these modifiers to act. So in this study, this is single cell. This is a within-person genotype-phenotype analysis, correlating the CAG length in a single cell. This is in the MSNs that exhibit very high levels of expansion to gene expression within individuals. And so what he's identified is 5 phases of the disease. A phase where the repeat expands at a slow rate for most for a very long time. Then it expands at a slightly higher rate in phase B, followed by phases C, D, and E of altered gene expression, C and D, and then elimination of the neuron. And so this essentially is a model to think about and might also provide different opportunities for those modifiers to act. So for example, a modifier could act in altering, say, a transition from a phase A to a phase B, or so on, as well as in toxicity phases downstream. Okay, so let me switch now to mice and work that we've been doing in the mice, and this has been a long time collaboration with Ricardo, who started this work in my lab, and we've sort of kept this going for for a while. And so having done a lot of genetic crosses in mice for a very long time, we developed this to speed up the process of identifying genetic modifiers and identifying new modifiers and testing modifiers, candidates as they come out from the GWAS. And so this is based on the knock-in Q111 line crossed with mice expressing, crossed with Cas9. And injecting AAVs, either PHPEB, which can target the brain, or AAV8 targeting the liver, and measuring the effect of knocking out specific genes on repeat instability in either the liver or the brain. The liver, I didn't say, exhibits very high rates of expansion, as does the striatum. It provides a really good readout for kind of screening modifiers. So we first tested this with known modifiers. So we knew that all of these modifiers, we'd done these experiments in constitutional knockout mice, that knocking them out stopped expansion. And so we validated this. So you can see we're injecting mice at 6 weeks of age, and then aging mice to 12 weeks of age. And you can see that anything that deviates from those kind of teal bars, that's the MT-treated mice. We have essentially, for those genes, knocking them out, suppressed expansion to that seen in 6-week mice. So these are very strong modifiers, and we can achieve this level of modification using this CRISPR knockout strategy. So then we went on to test a variety of other genes. We've probably tested over about 80 now, and this is 60 genes, and these are in various different pathways. DNA repair pathways is a big focus on mismatch repair, as well as candidates from the GWAS, and I'll highlight a few things on here. So the ones on the top, when you knock them out, they increase expansion, so normally they're suppressors. And then the ones on the bottom right there, underneath the line, when you knock them out, they decrease expansion, and so these are normally enhancers. And so this just highlights some of the stronger ones. I haven't put everything on here, but you can see all the mismatch repair genes. We also identified POLD1, POLD2, POLD3, POLD4, components of DNA polymerase delta. POLD1 was also more recently identified as a modifier of clinical phenotype in HD patients. And then on the left here, the suppressors, FAN1, we know that's a strong suppressor. PMS2, and we identified 2 new genes, HMGB1 and LIG4, as suppressors of repeat expansion. And so these things become potential targets, therapeutic targets, some better than others, for modifying the rate of expansion. Oh, the other thing to note, so these are genes that are also human modifiers. So LIG1, TSURG1, CCDC82, and RRM2B. And in this assay, this is in the liver, we did not find a strong effect of these on modifying instability. LIG1 is perhaps the most surprising because it seems very likely that it is involved, and we're looking at this some more, and I'll show you some more data on LIG1 in a second. So yeah, the other thing to point out is that we do see relative enrichment for mismatch repair pathways amongst all these modifiers. So many other DNA repair we haven't obviously done a screen and tested everything, but really, it's not just all DNA repair proteins that are involved. It does seem to be really coalescing around a specific pathway, which is the mismatch repair pathway, plus FAN1 is critical, not canonically a mismatch repair protein, but is clearly part of this process. The other sort of fun thing that's been interesting to do with this system is to test genetic interactions, because we can inject the mice with 2 AAVs, each targeting a different gene, and we can look at the impact there. So this is kind of complicated, and I won't go into details, but essentially, on the left, you can see single target knockout and the effects that we see on those genes there. And then you can see where we've targeted 2 different genes, like the 2 red dots would be 2 genes that are normally enhancers, and a red and a blue would be a gene that's an enhancer and a suppressor. So for the essentially all the enhancing genes, MSH2, MSH3, are critical, even in the absence of a suppressor that is promoting expansion. So really, this is sort of allowing us to start to dissect in a genetic way these different components of this process. And then the other thing, of course, is what about the brain? So we are expanding this now to doing some work in the brain, and this shows a number of the genes. When we target with, deliver with PHP-EB, which crosses the blood-brain barrier, and we can actually, this also goes to the liver, so we can actually look in the same mice in the striatum and the liver. And we see pretty parallel effects of modification in the striatum and liver. The interesting thing is PMS2 and FAN1, where knocking out FAN1 in the liver has a profound effect relative to knocking out PMS2, but the opposite is true in the striatum. So this kind of starts to speak to different tissue or cell type specific effects of these knockouts, which I think speaks to different protein complex and stoichiometries and different sensitivities to that. And again, thinking about the human data as well, where there may be cell type specific effects of modifier variants. So just to end, I'll just sort of briefly touch on some other thoughts, which is, so these are all knockout experiments. And so we're also interested in looking at functional variants in these proteins, because they can obviously provide sort of mechanistic insight. And then one, Ricardo, I think, will talk a bit more about this, which is in the MLH3 endonuclease domain. So we looked at a mutation that abrogates the endonuclease function of MLH3 and found that this is critical for expansion. So this starts to give us some more sort of functional insights. The other important thing, of course, is thinking about the human modifier variants, because that can tell us about some biology which is directly relevant to what's going on in patients. And so my lab is looking at some of these variants. The easiest ones are the ones that alter amino acid sequences, and so that they're relatively tractable to understand. So one of these that we're looking at is LIG1, and so there are several haplotypes of LIG1 in the human data, in the human modified data, but this is one of them. And it is a missense mutation, K845N mutation. It's very rare, but it has a very large effect on onset, on delaying onset. And so the idea is that if we can understand this, it may provide a route to having a large disease-modifying effect therapy. So together with In-Chi Song, at MGH, we're looking at this mutation in different ways, biochemically and in mice and patient cells, and trying to sort of dissect this mutation. And I'll go quickly through this. So this amino acid is conserved. We made a mutation in the mouse at position 843, and we have crossed those with our mice, our knock-in mice, and we've looked at the impact on repeat expansion, and we can see that this mutation in mice suppresses repeat expansion. in both striatum and the liver. So this is giving some evidence that in patients, this mutation may be delaying onset by suppressing expansion. So I'll end with a model that we've developed really based on the CRISPR work, but also this could incorporate LIG1, and also based on a lot of other data that has been generated in the field biochemically, and trying to put everything together the best we can. And so essentially the model is you have a CAG, CTG slip out, which can happen during some aspect of say transcription or DNA breathing or chromatin alterations. And this is bound by a complex of MSH2 and MSH3 called mut-S beta. This is critical. These are the critical components, I think, driving the expansion. This then recruits a complex called mut-L gamma, which is MLH1 and MLH3, as well as PMS1, and very little is known about this protein, but it's clearly involved in this process. And mut-L gamma cleaves the DNA, and it's been shown to cleave the DNA on the opposite side to the loop out, and that allows the loop out to be incorporated as an expansion. And we think that DNA pol D is really important in this process through strand displacement synthesis. And then essentially the ligase comes along and seals the nick and incorporates an expansion. So this is a model of the factors driving expansion. And we think that through mechanisms that are not fully understood, there are certainly hypotheses and models out there for FAN1 could interfere in this flow, if you like, and alter the chance of incorporating an expansion. And perhaps in different cell types, having different amounts of these proteins and protein complexes could make a cell more or less, give a cell a higher or lower propensity for expansion. And so then, sort of therapeutically, thinking about what we might want to do, going back to human again, and the modifiers. So this is a kind of cartoon depiction of neurons with repeats getting longer over time, getting to some threshold where they die, and eventually you get clinical onset. Onset delaying modifiers will slow that process of repeat expansion, and onset hastening modifiers will speed it up. And what we want to do is to try and find therapeutics that will slow, sort of turn to green, essentially. Slow or stop that process. So, and just to say, I think we'll hear more at this meeting from some companies working on this, but there are many companies now really exploring this idea and developing drugs targeting the process of somatic expansion by targeting these key modifier proteins. The other interesting thing, of course, is that HD is one of a family of repeat expansion disorders, some CAG, other triplets, and other flavors of repeats. So potentially, and many are characterized by having repeat expansion. And so this potentially provides an opportunity to intervene in many of these diseases by targeting the root mutation. And with that, I'll say thank you for your attention, to funders, patients, and families, and my lab and collaborators. We could do questions now, or we could do something at the end. Either way is good. Sure. There's a question on the chat. I can't hear what you're saying. Oh, yeah. So there's a question on the chat about measuring repeat expansions in living patients. So I think this is a key challenge in terms of therapeutics. in being able to find readouts of somatic expansion in cell types that make sense. So you can measure repeat expansion in blood. Darren Monkson's doing a lot of work on this, and it's certainly measurable. It's slow, and so that may provide an endpoint for if you're talking about therapeutics that are systemic. A further challenge would be for therapeutics that directly target the brain and needing readouts for that. So I think this is, and there's work going on to, for example, enrich exosomes from CSF and things like this. So I think that remains an ongoing opportunity and challenge, but yeah, important. Okay, Ricardo.
So thank you very much, Victor, and the organizers for inviting me. These are my disclosures. I'm definitely not an expert in what I'm going to be talking about. I'm much more of an expert on what Vanessa just presented, but I'll do my best. So I do collaborate with experts, so I've learned a few things by interacting with them. So this is the first time I'm here at Huntington Study Group meeting, so I wasn't quite sure of what the audience, so I apologize if some of the slides are too simplistic and some are too complex. So I'll try to navigate this as best as I can. But for the ones less familiar with CRISPR, so CRISPR is sort of bacteria's immune response system. They get infected by viruses, which inject their DNA. Bacteria sort of incorporate that DNA into these repeat structures that they then can transcribe and fuse with a Cas9 protein to go and destroy the viral genomes when they get infected again. So it's sort of a memory system for an adaptive immune system that bacteria have evolved. This was sort of engineered to work in mammalian systems. And here sort of describing the complex of the Cas9 protein with a guide RNA, so a small molecule of RNA that is complementary to your sequence of interest in the DNA. And then if this lands adjacent to a compatible PAM sequence, it will introduce a double-stranded cut into the DNA. If this happens, if you introduce this double-stranded cut, normally cells tend to repair this by non-homologous end joining. So they'll try to ligate these to loose pieces of DNA, and in the process, normally either remove a little bit of DNA or introduce a little bit of DNA, which we call indels. And these normally lead to frameshift mutations, and essentially your gene stops functioning. An alternative repair method is if you provide a donor DNA with the sequence that you want to insert, and it has some homology in the flanking regions, you can introduce, you can replace the sequence that was there with what you want. So you can correct a mutation and restore function of a gene. So Cas9, and I'm not going to go into all the engineering that has gone into this field, but essentially people have taken Cas9 that is cutting on both strands and engineered it in a way that it only cuts on one strand or it doesn't cut at all. If it cuts in one strand, we call it a nicase. If it doesn't cut at all, it's a dead Cas9. And other engineering experiments that have been done is you can fuse different functional elements to these Cas9 proteins. You can either add a transcription factor or a repressor to either activate, to recruit Cas9 and the transcription factor to the region of interest and either upregulate your gene of interest or repress it. You can also fuse it with DNA or histone modifying enzymes to either methylate the DNA or make modifications to the histones, again, to regulate the expression of these genes, of your gene of interest. More recent examples of tweaking this sort of toolbox is work done by David Liu at the Broad. It's called base editing. Essentially, you use a nickase, a Cas9 that doesn't cause double-strand cuts. And this Cas9 is fused to a deaminase. And with that, you can either target cytosines or adenine and sort of change just a precise nucleotide without having double-strand cuts, which is important. So here, sort of representing the two more common engineered variants where you can convert a C to a T or an A to a G. Another version or another engineering effort was to develop prime editing, also developed by David Liu. Here you fuse the Cas9 with a reverse transcriptase, and you use a guide that is sort of a two-in-one. It not only does it guide, but it also has the sequence that you want to introduce. So again, no double-strand cut. You just nick, you release the one piece of DNA, and then using the template, you can add the sequence that you want to introduce there or replace. And these are sort of safer strategies for gene editing, avoiding the double-stranded cuts. Now, CRISPR in the clinic. You're probably aware that just earlier this year, we've had the first FDA-approved CRISPR treatment for sickle cell. And this was sort of work in collaboration with CRISPR Therapeutics and Vertex. So quite remarkable that approximately or less than 10 years from the discovery of CRISPR to having an FDA-approved treatment. One thing I would say here is that, although this is exciting, this is still an ex vivo gene therapy. So essentially, blood stem cells are collected from the patient's CRISPR edited ex vivo and then infused back into the patients. And here are sort of, this is a recent paper from Fyodor Ornov, where he's sort of summarizing the landscape in clinical development of CRISPR therapeutics, and sort of listing here a number of companies that have active programs. The vast majority are sort of easy to reach targets, and it's expected some of them they're already in the clinic, but will have approvals in the coming years. I'll just touch on two of these highlighted here in red are in vivo editing. So the patients are actually injected with a CRISPR reagent, and the editing occurs in vivo. I'll highlight the case of from Intellia for TTR amyloidosis. And here they use a double cutter, so a normal CRISPR-Cas9, that they deliver in a lipid nanoparticle. And essentially, this disease is caused by a protein, the TTR, that is expressed in the liver and then misfolds in the brain. But so here they're knocking it out. They're using this CRISPR reagent to transduce hepatocytes, knock out this gene, and reduce this protein. And here's sort of the data from patients showing that even with low doses, you're getting 50% reduction of TTR, but this reduction gets us close to 100 with higher doses. And the important thing is that this is sustained. So these are permanent changes that occur. So you change the DNA of the liver cells, and these are permanent. And they are actively recruiting for a phase 3 trial. So, and here I'm not going to go into too much detail, but I'm sort of going to list what are some of the challenges. One, I would say it's delivery. Essentially, how do you get this CRISPR drug to the brain? So here I'm sort of focusing a little bit on the problem we have, which is HD. Some of the easier strategies would be using viruses, AAVs particularly. They have advantages and disadvantages, so I'll touch on a few. Disadvantages that you sort of have a one shot at this. You cannot read those with AAVs, especially if unless they're very different serotypes. A limitation that exists with AAVs is that normally they don't cross the blood-brain barrier. So if you give them systemically, they don't get to the brain. There are strategies, and you have the example of Unicure, where you can inject directly into the striatum. And there are ongoing efforts, and there are quite I'm mentioning here work from Ben Deverman, from Voyager, and others, where they've engineered these AAVs so that they can actually cross the blood-brain barrier and with a single systemic injection actually reach the brain. Problem with AAVs is that they have low capacity. So CRISPR proteins aren't extremely large, but they slightly exceed the capacity of an AAV. So if you have complex engineerings, you add other elements to your Cas9, it will exceed the capacity of your AAV, and you will likely need to use two. But there are other alternatives, and I'm listing here using RNPs or lipid nanoparticles, which is akin to what we all had with Moderna or Pfizer COVID vaccines. You can also package these into extracellular vesicles. So that's certainly a challenge, but I think we should be optimistic. There's definitely a lot of effort being put into addressing this issue. Like I said, a lot easier to do when dealing with the liver, when dealing with the eye, or but much harder when you want to reach the deep regions in the brain. Another concern is safety, and essentially what I mean by that is a CRISPR reagent doing what we want, or is it doing something else? One common problem with CRISPR is that you might have sequences that are either exactly the same or too similar to the region you're targeting, and Cas9 might be active there, so you might be making edits elsewhere in the genome. And again, these edits will be permanent. If these edits happen for example, in a tumor suppressor gene, you might cause cancer. So there's a lot of concerns, and you want to monitor very carefully, making sure that your drug is only editing your gene of interest and doesn't have too many off-targets. Another concern, especially using earlier versions of Cas9 where you're having double-strand cuts, you're creating stress within the cells if you're constantly expressing Cas9 and constantly cutting DNA. This might trigger DNA damage response, it might trigger chromosomal rearrangements, and have deleterious side effects. And again, depending on your delivery modality, you might be reaching into tissues that you may not be interested in expressing your Cas9. For example, germline edits are a concern for the FDA. So even though I've just showed you that list of companies actively pursuing these and having some of these reagents in the clinic, this isn't an extremely healthy space. There's a lot of companies right now that are in this space that are struggling to fundraise, and that number is still sparse. So the NIH has funded what they call Somatic cell genome editing program to try to fast track some of these novel therapeutics into the clinic. And one particular goal is in terms of optimizing genome editing-based therapeutics to support future clinical trials. With that, I'll list here the 5 grants that were awarded. These are quite sizable investment from the NIH. These grants are approximately $25 million each. And you'd be happy to learn that 2 of them actually include Huntington's disease as part of their program. So I'm going to go through a couple of these very briefly. The team led by Jennifer Doudna at Berkeley, sort of based on a strategy that was originally published by John Bin Lee and Bev Davidson, which sort of an allele selective excision of the repeats. And this is sort of enabled by the fact that you have SNPs that occur only in the mutant and expanded allele and not in the wild type. So there are combinations of SNPs that you could sort of target and make allele specific. But so they've taken this strategy and they're sort of pursuing it. Full details are not public, but these are what we can share at this point. But they're going with a two-guide strategy, flanking the repeat, one common, one SNP specific, to excise the expanded CAG only. They're testing a variety of different delivery systems, both RNPs and AAVs. And so they're funded to go and test these all the way from developed from in vitro all the way to tox studies and in non-human primates. The other project that I'm actually involved in, so this was spearheaded in David Liu's lab, and now it's being primarily led by Kat Lutz at the JAX. And this is sort of, the foundation of this is what Vanessa just presented, the fact that interruptions seem to modify the age of onset. So, and we believe that this is likely due to the reduced rate of somatic extensions. When you have an interruption, the repeat expands slower. So this leads you to hypothesize that could you add interruptions to the repeat to stabilize it and delay onset? So Zanetta, a graduate student at the Liu Lab, has sort of been playing around with this and sort of developing base editing strategies targeting the repeat itself. So you're sort of going to have multiple landing spots for your CRISPR reagent within the repeat, but with the idea that if you introduce, if you change the CAG into a CAA, which still codes for a glutamine, so you're not changing the protein, but you're interrupting it, making it more stable. At least that was the hypothesis. So here she's testing this in patient fibroblasts, and you can see that she's having quite strong editing both in the, so this is non-allele selective. You will end up targeting both the wild type and the mutant allele. You have obviously more edits in the mutant allele, because you have sort of a larger landing pad for the Cas9 to interact with. And again, going back to the sorry, to the limitations we have with these reagents is that they're large. So we're packaging them into 2 AAVs, and here we're using AAV9, and we're injecting in neonates by ICV. And then we're looking at instability, the degree of interruption and instability at 12 and 24 weeks. And this is also done in the QN11 mice. And here we're showing the degree of editing. So not super high, but around 30, 40%. That's the maximum we can achieve in the cortex at 24 weeks. Slightly lower in the striatum, but somewhere between 20 and 30%. But remarkably, we can show that even these low editing efficiency is enough to slow down significantly the rate of expansion, and surprisingly, in the striatum, even promote contractions. So we're quite excited about this, and this is ongoing. Like I said, we're conducting more thorough preclinical studies at the JACs. Yeah, so I think I mentioned some of these, but sort of reiterate that it's non-allele selective. You're targeting both alleles, but you're converting CAGs into CAAs, exactly coding to polyglutamine. The protein should remain unaltered. Touch on the delivery. Yes, off target. So this is targeting the CAG. Naturally, there's multiple CAG stretches throughout the genome. So that will be something that we'll need to pay closer attention to. And like I said, we're conducting studies at JACS now. And interestingly, John Min and Vanessa was involved in this study as well, have sort of spearheaded a similar strategy, and that is published now, if you want to go check that. I believe that in vivo editing, they don't have as much data, but sort of supporting the fact that it can be done. And now I'm sort of going to touch on a few, sort of skim the literature and looking for what are other people trying to do, what are alternative strategies for using CRISPR gene editing in the context of HD. And this is one of the earlier papers showing that you could use a double cutter and just flank the repeat and reduce the level of mutant Huntington aggregation in the brain of, I forget what mouse model this was. I think this was Q140, if I'm not mistaken. But a problem here is that you're non-allele selective, so you will be knocking out both copies of the Huntington gene. You'll eliminate both the wild type and the mutant Huntington protein. And that goes sort of to the discussion earlier this morning, which I won't get into. An alternative strategy here is based on the idea that caspase-6 cleavage in exon 13 is sort of important driver of disease. And here this group is using a base editor to target just upstream of exon 13, excuse me, to sort of promote splice shifting and eliminate this caspase cleavage site. Again, non-allele selective. This sequence will be common across both alleles, and it may not address the issues such as HTT1A. Here's another strategy, sort of going back to some of the data that Blair presented this morning, where you can recruit, or here there's multiple steps in this study. They sort of mapped the promoter region of Huntington gene, and then they sort of introduced base editing mutations in the same site that Blair mentioned earlier today to sort of reduce the levels of Huntington. Again, this will be non-allele selective, and it will result in permanent reduction of both alleles. Another study led by Vincent Dion in Cardiff leverages a nickase targeting the Cas9 repeat. So what is shown is that by tiling also using a CAG targeting guide and an enzyme that doesn't cause double strands, it only cuts multiple times on a single strand, that this results in contractions. And they had done that originally in cell lines, and he's just got a preprint out now where they've done that in vivo, where they actually in the same mouse inject both on one side just the nickase and the other side the nickase and the guide, the CAG targeting guide, and they show that specifically on the striatum where the guide was included, they are seeing a high degree of contractions, which I don't know if you can see here. I'm trying to point. Sort of here. So you see a reduction of expansions and a promotion of contractions, which is quite exciting. And now I'm going to sort of touch a little bit on what are we doing in this space as well, targeting somatic instability, and sort of why we asked to sort of merge these two sessions, because it sort of follows well from what Vanessa presented. So that was the model, and I can show you, go back. So as you can see here, MLH3 seems to play an important role here. We know that the endonuclease activity is critical for this process. And why do we know that? We performed an experiment where a mouse had a single point mutation in the endonuclease domain of MLH3 that inactivated. It still expressed MLH3, but it inactivated its catalytic activity. So it could not cut the DNA. We crossed it in with a Q11 mice, and what you can see is that once you have a homozygous mouse with that mutation, you absolutely stabilize the repeat. You have no expansions whatsoever, no matter how long you age the animal for. And that is seen across all tissues. We, at the time, also collaborated with Ed Grabzik, who was developing splice switching oligonucleotides for these exon 7, which is where the endonuclease domain is expressed or coded in MLH3. And with that, so this gene can be expressed in two different flavors, the variant 1 with the endonuclease domain and variant 2 without. And in fact, we looked into GTEx data. In some tissues, this is a naturally occurring variant. In some tissues, you even express more of variant 2 than variant 1. So we know that variant 2 is not sort of a deleterious byproduct of that splice switching intervention. And so we treated both. So we started by treating HD patient fibroblasts with this ASO. And we could see that we could convert the splicing of MLH3 to the endonuclease lacking variant, and we strongly suppressed CAG expansions in this line of HD fibroblasts. We tried doing this in vivo in Q11 mice, but I should say that these weren't very well designed ASOs. They were quite toxic, so we couldn't. We had to do very frequent dosing, and we couldn't increase the dose very high. So we only managed to splice switch the MLH3 in the kidney because the ASO was being so quickly cleared through the kidney. And there we saw a significant reduction in somatic expansion. So we thought that this would be a viable strategy for addressing somatic expansions in HD, but we weren't too happy with an ASO strategy, so we sort of turned towards CRISPR for this. And the idea behind that was that the splice acceptor is extremely well conserved across species. So we knew that if we were to disrupt this AG sequence, we would be able to alter the splicing and splice exclude this endonuclease domain containing exon. And so for that, we collaborated with David Liu using base editors like I mentioned, that have the ability to, without cutting the DNA, they just nick it, but they make a single base change. They can convert an A to a G or a C to a T. And the other aspect of, which has been a very, something that the field has focused a lot, that I didn't touch on before, but the PAM is quite restricted. The fact that CRISPR, or regular CRISPR, needs an NGG sequence for Cas9 to work, it's sort of restricted, and it only allows you to go to certain places in the genome. Ben Kleinsteiber has sort of engineered these Cas9s in a way that they sort of can go and recognize different PAM sequences. So we combine these two sort of advancements and sort of tile the region of interest with all possible guides and a number of different base editors. And sort of here showing a heat map in terms of the editing efficiency. And as you can see, so ABE1, sorry, I didn't label this, ABE1 is actually the wild type Cas9. So this wouldn't have worked if we had just used the wild type. But when we use SPG, there's a couple of guides that have very specific editing in the A nucleotide that we're targeting. When we use this in hex cells, we can show that if we edit both alleles, we completely shift the editing of the sort of bad variant, the one that contains the endonuclease, which is the upper band, and we only express the variant at the bottom, which does not have the endonuclease domain. And more interesting, when we use a cell line that has time-dependent CAG expansions, you can see here this plot with increasing CAG. Sorry, had a cough last week. The CAG goes up across these 28 days, but when we mutate, when we use our base editors and we edit the splice acceptor, we completely stabilize the CAG repeat. And this is after 4 weeks. We've now gone all the way up to 30 weeks. And I would say that the bottom trace is still the same. The top, the unedited has sort of left the slide. It's expanded significantly. So this is something that we're still actively working on. We've started testing this in mice, injecting them, packaging them into AAVs. We've had to humanize the mouse. So that's another issue with CRISPR strategies, is that you develop them against human sequence. And if you want to conduct preclinical testing, often that sequence is not present in your mouse model, so you'll need to generate a humanized mouse model. And we're also testing this in patient-derived cellular models. Again, the same important considerations I mentioned, which is in terms of safety, we want to make sure that we're only editing this gene and not others, at least not significantly. And if we're editing other genes, we need to make sure, or other places in the genome, we need to make sure that they're not coding important genes, and they're, we've done some preliminary analysis into this, and we're primarily targeting regions in between genes, so we're not too concerned. And the other issue is we're using AAVs. Is that going to be a viable delivery strategy? We don't know, but that's sort of the best strategy we have right now for conducting these preclinical testings. And yeah, so the sort of advantage of these strategies, they're sort of a one-and-done therapeutic strategy. You treat with this drug, you make a permanent change into the DNA that's sort of sustained throughout the life of the patient. And another thing that Vanessa sort of touched on, with this strategy, we're targeting an upstream mechanism, something that is common between different repeat expansion diseases. So we might be developing a therapeutic strategy that will have multiple applications. And with that, I want to thank the HDF for awarding us a transformative research award, and sort of listing here all the collaborators that are involved in this study. And I believe with that, I'll take any questions. And I'm sorry for the coughing. I don't know if we have mics.
You go.
Hi, thank you both for some really amazing talks. I'm just curious, as both experts in
come up?
somatic expansion, the current thinking in your field of the possibility of the expanded CAG repeat in the DNA causing HD as opposed to the protein itself. So the actual expansion of the CAG being toxic compared to the protein itself.
I don't think that's really what we're proposing. It's just this hypothesis does not eliminate the fact that Huntington protein might be the toxic driver, right? If you have a longer CAG repeat, you're going to have a longer polyglutamine expansion, right? So I want to make sure that the message of that paper is not saying that it's all about the CAG. It's not the DNA that is driving toxicity. That's not what it's being proposed. Could be a variety of other mechanisms, which could include protein. If I understood it correct.
No? Yep. So just to add to that, the somatic instability, that's driving the rate at which you get to a toxic species. And then the really recent data from Steve McCarroll's lab might suggest that you have to get to quite a high repeat length. He's estimating 140, 150 for toxicity to occur. That's that model, but it's not the repeat expansion per se that's toxic. It's just that the fact that it has a propensity to expand, and the rate at which you have to get to some threshold, which then triggers toxicity. And the toxicity question is open, I think, as to what actually is that driving. Is it driving Huntington 1A? Is it driving something at the level of the full-length protein or RNA? You know, so there's many different potential modes of toxicity. It may be different in different cell types, but yeah.
Hi. Thank you for those excellent talks. There's a question for Ricardo. So if a prime editing approach was feasible, would there be an ideal number of CAG or CAA repeats you would rewrite in? And is there a lower limit? What would happen if you do 0 or remove it all or 1?
Yeah, I don't think we know what the lower limit is. I think most strategies that are being experimented with actually result in that, in sort of a complete loss of the CAG. I think a colleague in our group has recently looked into the UK Biobank database to sort of try to understand exactly what's the smallest CAG that exists. I can't remember off the top of my head, but it's quite short. There are individuals going around with very short CAGs. They're rare events, but I can't remember the exact number.
Thank you.
Thanks so much for the talks. So maybe I misheard, but I think Vanessa said the CAA, CAG duplication, that delayed onset, and then when it was not there, hastened onset, was not at the level of somatic expansion of regulating that. But Ricardo's telling us that you're going to change that. You're going to add more CAAs to affect somatic expansion or impact. So I was a bit
the first part?
confused.
Yeah, so the pure CAG length is the key driver of somatic expansion. If you make that shorter by base editing, then you will reduce the rate of expansion. So that's the therapeutic strategy by introducing the edits, right? So you will shorten the pure or any pure repeat length. So that's, but in the human alleles, if you then take, for the same pure repeat length, so you have 40, and then you have the canonical N, CAA, CAG, or you have 40 plus the duplication, then the question is, what's the difference between those two? And in those, or when you don't have the CAA. So if for the same pure repeat length, then so far there isn't any good evidence that, if you can think about it as the addition of those and bits onto the end. The different ends for the same pure repeat length do not, at the moment, from what has been looked at, seem to act by altering the rate of expansion. So does that make sense?
It does. So could you just insert CAAs without even changing the CAG? That would, I guess, be the experiment to test whether interrupting with the CAA makes any difference.
Introducing CAAs just
without
yeah.
changing CAG, just to further test what you think you know from the human. Because I guess I'm confused. It still produces polyglutamine, but it doesn't affect somatic expansion. So
But
yeah.
So you can think of it as the flanking sequence. If you have the same pure CAG, what's the flanking sequence doing? So that could be acting in different ways. That could be altering expansion. So far, at least from the data that's available in many patients, that's in blood, it doesn't seem to be. It doesn't rule out that that could be a mechanism in brain. But there's very few samples with, right? So, or is it a flanking sequence doing something different? And that could be altering, aggregation. It could be altering HTT1A production. It could be altering RAND translation. I mean, there could be lots of things that flanking sequence is doing.
Yeah, but there's evidence that putting these interruptions in the middle of the repeat, you have a very strong effect in stabilizing. And CHDI, and I think this is now in John Min's paper, CHDI generated a mouse model with severely interrupted polyglutamine, so CAG, CAA, CAG, CAA, very heavily interrupted, and those mice have absolutely no somatic expansions. And we haven't gotten there yet, but I think once we start looking into our data in a different way and start looking at neurons where we've edited more strongly, where we have more interruptions, and measuring the somatic instability specifically in that neuron, I think we're going to learn a lot more about the relationship between the degree of interruption and the impact it has on somatic instability. All the data that I presented is sort of done in bulk. Like I said, we only had something like 20% editing, so only 20% of the neurons actually had interruptions, some degree of interruptions, but that was still enough to see reduced expansions and promotion of contractions. And don't ask me how we're getting the contractions, I'm not entirely sure. So this is something new that we're observing specifically in the striatum. We don't see it in the liver, for example. So mechanism, I'm not quite sure.
I was just going to follow up Ricardo over here. I think William Yang also, and many of the transgenic animals that were generated had interruptions in their repeats and didn't have much instability. And for a long time, we didn't understand why that was. But it was because they weren't pure repeats. And now he's generated a back that has a pure repeat and has instability compared to the interrupted back. So I think that's another line of evidence that in vivo suggests that instability is related to the pureness of the CAG.
Those weren't that heavily interrupted, if I remember correctly. Like the Yak 128, I think, had something like 3 interruptions.
That's correct. Middle. It doesn't take a lot of interruption of the pure repeat to have an effect, I believe.
But they were sort of in the middle.
Hi, David Margolin from Unicure. At the recent Wexler Symposium, there was data presented that goes to the question about how the repeat length expansion might relate to disease onset. So firstly, there was evidence increasingly showing that the HTT1A alternative splicing fragment is important in the pathogenesis. And secondly, and relatedly, that there's a preferential production of that alternative splice form, the HTT1A form, when the repeat length gets into the same 140, 150 expansion length identified by the Macaro lab as being triggering the onset of toxicity in neurons. So potentially the somatic length instability reaching this critical threshold of around 140, 150 increases the production of the particularly highly toxic HTT1A alternative splice form.
My question is, if you start off in life with like 41 CAG, and due to somatic expansion, in the striatum it hits, let's say, 100, which is maybe the threshold where neurons start to die. How do you test from a live human being what your, can you test that? How do you test that, I guess is my question. Like in a brain, like in a live human being, how can you tell what your CAG count is in the striatum right now?
Not easily. Yeah, I would be great. You obviously can't biopsy the brain. So what's been done now is on postmortem. So we don't know exactly what the repeat length is in the neurons at onset. So I think that's obviously challenging. I mean, maybe some of these, PET ligands that could use, aggregation as a readout of that may be useful in that. But yeah, quite difficult. I mean, a lot of modeling is going on, I think, to try and to model that and, put it together with clinical data. Some of Steve McCarroll's modeling would be nice to see put together with the clinical data. Yeah.
Isn't it true, though, that one neuron, let's say if it's at 100, that the neuron right next to it in the striatum could also be like 120 or 80, it could be very different, correct?
Yeah, so the idea is that the repeat starts to expand, sort of, and they asynchronously, and each neuron is doing its own thing, and the repeat is, so at any one moment in time, you have a kind of cross-section where you have a mosaic in the brain, and all the neurons have different repeat lengths. And some may have 500, some 100, some 40, just depending on the rate in those. And as you go on, you push it towards longer and longer. But yes, it's effectively a mosaic.
Okay, thank you.
But going back to the notion of biomarkers, like Vanessa presented some data, but that's only from 3 patients, where you correlate the degree of instability in blood, CSF, or liver, sort of tissues that might be accessible, and they strongly correlate with the degree of instability that you observe in the brain. I think if that was to be done with larger number of samples, that you might be able to model that. I think it's very limited what we can assume based on just that study we did.
I mean, the rate may correlate, but the question is about what is the actual repeat length in the neurons at onset.
Yeah, which you might be able to model, right? If you have data from postmortem material and with matching blood, with matching CSF, with matching liver, and you sort of have enough patients at different levels of somatic instability that you might be able to model by taking from a living patient, taking a blood sample, and they get a certain degree of somatic expansion that you'd be able to sort of predict what it might be in the brain, and potentially at some point factor in their genetic modifiers, like typing and sort of understanding is this an individual that is more likely or less likely to expand. But I think we need just larger sets of data for that.
Thank you guys for a wonderful presentation. I wanted to, are there any more questions from anybody? I wanted to close by asking you each to crystal ball it as much as you can. Obviously we don't know these things, but the questions that I'd like you guys to close with is if you had to predict how, some people have said somatic instability is going to be a driver of disease progression earlier in the disease phase, and maybe it's mutant Huntington that's driver of progression of the disease later in the disease, or how somatic instability overall will pan out in what's the toxicity mechanism of HSG. What would be your guess at this point? And then also, what would you guess would be the delivery solution for therapies like CRISPR? Would it be the dual AAV approach or things like that? As things stand now, obviously impossible to predict, and we're not going to hold you to it, but what would you throw out there for both of those?
Sure. going back to the first question, I think somatic instability is an ongoing driver of disease in different cells, right? So it's constantly happening, and it's constantly pushing those neurons to dysfunction. And they're all sort of expanding at different rates. It starts in the striatum. Eventually, you have neurons in the cortex expanding as well. So I don't think that two-phase is sort of very for that specific cell. So, and they're all sort of going through that path simultaneously at their own rate. Do you want to add anything to that, or?
Yeah, I just come back to the human data and what the human genetics tells us, and that the strongest thing it tells us at the moment is that the somatic expansion, or the modifiers of that process, can alter onset. They can alter the rate at which you get to a toxic process that then gives you clinical onset. So I think in terms of where that sits, and there are other modifiers, we don't know what they do, but this is the strongest signal that there is from, an experiment in people with naturally occurring variation. It's altering the rate of onset. And so I think there's great hope that modifying that process can slow that before you even get to onset. I don't think that precludes other things, lowering as well. But this is where the strongest data that we have exists from humans, from patients. And I think there are multiple modes of delivery, potentially, right? So.
Yeah, I guess it depends a lot on what's your therapeutic modality. Are you going to lower the levels of MSH3, for example? Different genes might have different thresholds for how much they need to be lowered to have an effect. For example, some of the data that I was presenting, we're completely stopping expansions, which might be different than with a lowering strategy where you might just be reducing the rate of expansion. So in an individual cell where these edits are made, you completely stabilize it throughout life. So I guess it will depend a lot on what's the mechanism of your drug, how do you deliver it, what's the percentage of neurons that you actually target with that. So I think that there's some strategy, reducing it by 50% in a large number of neurons might be better than reducing it by 100% in just a lot less neurons. All of these strategies are sort of evolving. For the gene editing approaches that I was talking about, I think we're going to learn a lot in the next few years in terms of delivery. I think that delivery is really the key thing. And I think the first strategies will involve good AAVs that will spread well and transduce a large number of neurons initially in the striatum where it starts. But eventually you need to get to the whole brain, I think. So that you might start off with an AAV strategy and then come back to the same individual and top it up with a lipid nanoparticle, for example, for the same gene, for the same sort of cargo, but delivered in a different way, and sort of top it up, give it to different parts of the brain as needed.